Hello students, welcome back to your own channel Vedantu NEET English. This is Baswaraj sir, your biology master teacher. Students, before we start today's amazing class, quickly let me know in the chat right now if I am audible and the beautiful presentation behind me is visible to each and every one of you. Good afternoon, good afternoon, good afternoon everyone. Now students, this is a part 3, right? This is a part 3 of the cell, the unit of life. Now, in the previous part, if you have not watched the first two parts, I would highly recommend watching the first two parts because it is very important for you to build up that connection, right? See, if there are 3 parts to a movie, you will obviously watch the first 2 parts. Third part will come later on, right? So, if you want to understand the third part better, because we will be doing a lot of questions. That's right. Today's class, PYQ session also is there. So, after we finish the syllabus, after we finish the chapter, we have a lots of PYQ questions. So let me know in the chat if you're excited for PYQ questions also, because we'll be solving a lots of PYQs today. Now students, in the previous class, we finished with endomembrane system, right? We did endoplasmic reticulum. We finished Golgi complex, Amazon factory. After that, we did lysozymes. We also did vacuoles. Done. Apart from that, we also did the double membrane structures, that is your Golgi complex, and also your mitochondria. Not Golgi complex, the mitochondria and chloroplast. Mitochondria and chloroplast, double membrane structures. So all that is over now. All that is over. Now what is left now? What is left? The left part is going to be ribosomes, the structure of cilia and flagella. Students, tell me in the chat honestly, how many of you think the structure of cilia and flagella is difficult? Let me know in the chat. Okay, because you need to understand what is this microtubule, what is this 9+0 arrangement and 9+2 arrangement. All that doubts will be cleared in today's class. After we'll be doing the structure of nucleus, chromatin, and finally the chromosomes. After that, PYQ sessions. So students, show some energy in the chat. Because when the line by line series started, I used to see so much of Josh, and all of you were super excited to learn line by line. Yes, sir, we will learn line by line of NCERT. We will crack NEET 2025. But where is that Josh now? Where is that Josh? Show me a Josh now. Where is that Josh which showed that, yes, sir, if you teach line by line, I will learn line by line? Where is that energy? Show us that energy, because we feel from your side it is decreasing day by day. So be serious. Understand with me, right? Understand with me. Now students, let's read the ribosomes first. It is going to be very easy chapter, very easy chapter. So one by one we learn. First, let's understand with ribosomes. So quickly let me know in the chat, do you find ribosomes? Do you find ribosomes in the case of prokaryotes true or false? Quickly. Do you find prokaryotes? Do you find ribosomes in the case of prokaryotes true or false? Quickly let me know in the chat. Obviously, sir, ribosomes are found in the case of prokaryotes as well as eukaryotes, both the organisms, both. Okay. Now, ribosomes are granular structure first observed under the electron microscope as a dense particles by George Paldane. So, George Paldane was a scientist who observed the ribosomes under the electron microscope. Now, tell me, do you find ribosomes? Can you see ribosomes under normal compound microscope? True or false? Quickly go. Simple questions, everyone. Can you see the ribosomes under a simple compound microscope? True or false? Quickly. Now, they are composed of ribonucleic acids. They're made up of RNA. So if they ask you a question, what is ribosomes made up of? They're made up of RNA and protein. RNA plus protein is the composition of ribosomes and are not surrounded by any membrane. Very important line. So I can make a question here. Okay. Ribosomes have how many layers around them? A single layer, 2 layers, no layer at all, both A and B. What is your answer? Tell me, what is your answer? Students, simple question. Ribosomes is covered by how many layers around it? 1 layer, 2 layer, no layer at all, no membrane bound, or A and B, both A and B. Quickly go in the chat. Okay. The eukaryotic ribosomes are ATS ribosomes, that is eukaryotic cell. All of you should know this. ATS in the case of ATS in the case of ATS in the case of eukaryotes. ATS in the case of eukaryotes. In the case of eukaryotes. Whereas 70S, which you learned already, 70S in the case of prokaryotes. In the case of prokaryotes. Remember that. Remember that point. Students, again, important point to be noted here is what is ribosomes made up of? What is ribosomes made up of? RNA plus protein. RNA plus protein. Along with that, how many membrane, students? Membrane. How many membrane, students? No membrane. No membrane. No membrane. Very important to understand. Very important to know that there is no membrane when it comes to ribosomes. Very good. Very good. Next now. Next. Let's go to the next concept. Now, prokaryotic ribosome is 70S. Now, each ribosome has two subunits. One large and one smaller subunit. The two subunits of ATS ribosomes are 60S plus 40S. Students, don't tell me later on, sir, 60S plus 40S is 100, but why 80? Because this is based on the sedimentation coefficient. This subunit is sedimenting at 60. One more subunit is sedimenting at 40. Together they are sedimenting at 80. Understand? That is sedimentation coefficient. It is not additive. This is biology. This is biology. Okay. While 70S ribosomes are 50S and 30S. Now, what does the S stands for, students? Question is here. Again, question is very simple. The S stands for Sedgwick unit. Sedgwick unit. Trisha, it does not have any membrane. Can you see? Membrane is no membrane. Is not surrounded by any membrane. It does not have any membrane. That is why ribosomes can also be found in prokaryotes. Because we know in the case of prokaryotes, the membrane bound organelles are absent. So ribosomes is also present there. Right? So they are not surrounded by any membrane. Beautiful. Nice. Now, Sedgwick unit stands for sedimentation coefficient also. Okay. Sedimentation coefficient. It is indirectly a measure of density and size. Very important. So what is the meaning of sedimentation coefficient, students? What is the meaning of sedimentation coefficient? The meaning of sedimentation coefficient is that if this is a beaker right here, students. Oh, you became very bright, huh? Okay. So students, if this is a beaker, if you can see a beaker here and we are adding the ribosomes here. So we have one small ribosomes, one small subunit, a large subunit. When you add them together, they're ending up around ATS somewhere here. ATS. When you add the smaller subunit only, it is sedimenting somewhere here. Sedimenting somewhere here. That is around 40S. When you add the largest subunit, it is somewhere here. That is 60S. That is sedimentation coefficient. When you sediment the ribosomes, where are we finding them? Where are we finding them? That is called as sedimentation coefficient. Now, what is the measure here? The measure is density. Indirectly a measure of density and size. So, can I tell the 60S ribosome, 60S subunit is much more larger in size compared to 40S? Yes or no in the chat quickly? Can I say 60S subunit is much more larger compared to 40S? Yes or no quickly? It's much more larger in size and more density also. Quickly. Yes, students. Now, both 70S and 80S ribosomes are composed of two subunits. Okay. See here. You can see his. Ribosomes in prokaryotes are small subunit is 30S, large subunit is 50S. Together they are called as 70S. While in the case of eukaryotes, small one is 40S, large one is 60S. Together they are called as 80S ribosome. Easy. You also understand ribosomes. There's nothing difficult here. The main points are already given here. These are the main points. Apart from that, these are the main points. That's it. A ribosome concept is over. Now, students, this is one concept called as cytoskeleton. There is a concept called as cytoskeleton. Now, if you by heart it, you can by heart it very easily. But if you're my student, I want you to understand what is cytoskeleton. Okay. Now, how we have a skeleton? Like our body has a skeleton, that is the bones. A body is made up of bones. Similarly, in the case of cell, it also has a skeleton that is called as cytoskeleton. Clear? Like how our bones is made up of calcium, we have bones. In the case of cell, we have three different types of cytoskeleton. Nature. Each one is very important, so understand first. There are three types of cytoskeletons here. First one is called as microtubules. Understand, students? There are three types of cytoskeletons inside. Three types. There are three types of cytoskeletons found inside the cell. Three types of cytoskeleton. Okay. Three types of cytoskeleton. First one is called as microtubules. Microtubules, which is very important, which we'll come back to later on. Then we have microfilaments. Microtubules, microfilaments, and intermediate filaments. Last one is intermediated filaments. The third one is called as intermediate filaments. Okay, nice. Intermediate filaments. Now students, understand what is it? Microtubules. Microtubules are said to be hollow. They are said to be hollow in nature. They are going to be hollow in nature. While your microfilaments are much more solid in nature. Even your intermediate filaments are hollow in nature. They are much more hollow in nature. That is from the inside. When you look from the inside, they'll be hollow. They'll be hollow in nature. Okay. Now, microtubules. Microtubules are mainly made up of tubulin protein. They're made up of tubulin protein. What about microfilaments? Microfilaments are made up of actin and myosin. Actin as well as myosin. Intermediate filaments are made up of some type of non-contractile proteins. They're made up of non-contractile proteins. Okay. So they are non-contractile in nature. Now students, you tell me, this actin and myosin, have you heard of this somewhere? Actin and myosin. You might have heard about this somewhere, actin and myosin. If you remember the chapter, the locomotion, actin and myosin is involved in the contraction of muscle. Yes. So microfilaments are made up of actin and myosin. They have contractile properties. They have contractile properties. While your microtubules are non-contractile. They're going to be completely non-contractile in nature. They're going to be non-contractile in nature. So if they are non-contractile in nature, students, what can they do? They can form assembly. They can assemble themselves. Like Avengers assemble, no? They can assemble themselves. I'll give you a simple difference. I'll give you a simple example, you'll understand. Microtubules example, the best example which you'll be studying in this class also, in your chapter also, that is going to be cilia and flagella. Cilia and flagella and centrioles as well as centrioles are made up of. Cilia, flagella and centrioles are made up of microtubules. They are made up of microtubules. Clear everyone? They are made up of microtubules. Let's read it what's given here. This much of extra information is required for you to understand the structure of cilia and flagella. Because when you see the structure from top view, you can see they are hollow. They are made up of microtubules. Understand the concept of microtubules right now, because this concept of microtubules will be used later on when we understand the structure of cilia and flagella. Clear everyone? Now, an elaborate network of filamentous proteins. See, there are filamentous proteinaceous structures consisting of microtubules. So all of them made up of what? All of them are made up of protein, actin and myosin, non-contractile protein, tubulin protein. So all of them are made up of proteins, filamentous proteins of microtubules, microfilaments and intermediate filaments present in cytoplasm in a collectively referred to as cytoskeleton. So microtubules, microfilaments, intermediated filaments, together all three of them are called as cytoskeleton. They're called as cytoskeleton. The next question arises now. What is their function? What is the function of cytoskeleton? Understand now. The function of cytoskeleton is that here. Mechanical support, obviously. If they are cytoskeleton, just like your bones, obviously they need to provide support, structural support. Yes, structural support. Motility. They help in cell motility. They help in motility. Very important. Remember this. Very important. Very important. Maintenance of shape of the cell. Maintenance of shape of the cell. So there are three main mechanisms. First one is motility. Then we have support and maintenance of the shape of the cell. Any doubt in cytoskeleton? Quickly ask me. Any doubt here? Quickly ask me. Paul, Rudranath, Ravindrani, Meghna, Jasli, Madhumita. Quickly ask me the doubt. Any doubt here? This is the information which you need to know about cytoskeleton. Information about cytoskeleton. Okay. Any doubt? You quickly ask me. Any doubt? You quickly ask me, students. Quickly ask me. No doubt? If there is no doubt, now we will go to cilia and flagella. Now we will go to cilia and flagella. Now, students, you need to understand the first main point. The first main point about cilia and flagella is that the flagella structure in the prokaryotes, the flagella structure in eukaryotes, both of them are different. Structurally, they are very different. Structurally, the structure of flagella in prokaryotes and eukaryotes is different. Now, I will draw the structure of flagella. All of you, I want you to draw with me for you to understand the concept. If you just see this video, useless. Draw with me, then only you will understand the structure of flagella. Okay, structure of flagella. Draw with me. Draw the structure of flagella, all of you. Draw the structure of flagella. Okay. In the center, we have two microtubules. In the center, we have two microtubules, which are connected by a bridge. Two central microtubules. Around them, around the two central microtubules, there is a sheath. There is something called a central sheath. Central sheath is present. Central sheath is present here. Okay. Central sheath is there. Now, from the central sheath, you can see 9 radial spokes. You can see 1, 2, 3, 4, 5, 6, 7, 8, 9. In total of 9 radial spokes are coming out from the central sheath. Okay, 9 radial spokes are coming out of the central sheath. Okay. Now, at the end of each radial spoke, there is peripheral microtubules. There is doublet of microtubules. Okay. A, B. Okay. There is something called as linker. Okay, B12 linker is there. Now students understand, there is also dynein arms. We can draw the dynein arms. It is not in your syllabus, but let's try to draw it. Outer dynein, inner dynein. Now, students understand, here we have the entire structure of your cilia, flagella is made up of microtubules. It is made up of microtubules. Now here, can you see how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. There are in total of 9 peripheral microtubules. Peripheral microtubules are there. Peripheral microtubules. That is, and they're present in the form of doublets. They're present in the form of doublets. 9 peripheral microtubules which are present in the form of doublets. Okay. Now in the center, do we have? Yes, we have two central. We have two central microtubules. Two central microtubules are there. There are two central microtubules. The central microtubules are connected to the outer microtubules with the help of radial spoke. Radial spoke is never directly connected to the central microtubules. Radial spokes are always connected to this regular part. Can you see this regular part? That is called as central sheath. Central sheath. This is the structure. This is the structure of your entire structure of cilia, flagella. Now here we can say there are totally 9 microtubules on the periphery. Yes. Plus 2 in the center. Yes. Plus 2 in the center. So this is a 9 plus 2 arrangement. Where we have 9 peripheral microtubules, 2 central microtubules. Periphery are connected to the central with the help of radial spoke. Radial spoke is connected to the central sheath. In the outer we have each peripheral microtubules is connected to each other with the help of BA linker. Linker are there. Linkers are able to connect the two different peripheral microtubules. Two different peripheral microtubules are connected to each other with the help of BA linker. Can you hear this purple color thing? This is called as a dynein arm. They are called as dynein arms. In the center, this part is called as bridge. This is called as the central bridge. That's all. Did all of you write down? Let's read what is given now. Let's read what is given here. See the diagram one more time. This is called as your, see here students, we have in total of 9 peripheral microtubules. Can you see? 9 peripheral microtubules which are present in the form of doublets. Okay. Outer layer is called as plasma membrane cover. Yes. Here we have central sheath. Here we have radial spoke which is connecting the peripheral microtubules to the central one. Peripheral microtubule to the central microtubules. Here we have interdoublet bridge, that is basically your type of your linker. Here we have the central myotubules. Here we have the central myotubules. This is how it looks. This is how it looks in under electron microscope. Under electron microscope. Done. See, very easy to understand. This entire thing is called as the core, and other name for core. The other name for this entire core is called as axoneme. It is also called as axoneme. It's called as axoneme. Let's read here. Cilia and flagella. Okay, let's do flagella are hair-like outgrowths of the cell membrane. They are the cell membrane. Now here you should also write the difference also. Let's write the difference also. Very important to understand the difference between cilia and flagella. Okay, let's write down cilia and flagella. Cilia and flagella. Write down. Okay, let's read one more time. Cilia are small structures which work like a orb. They are going to be small structures. They are small structure which work like orb. All of you know what is orbs? All of you know what is orbs? Orbs are basically that movement. Like, if you're sitting in a boat, you try to move the boat. That is called as orb. See here. This is called as an orb. This is called as an orb. Okay. It's called as an orb. Or causing the movement of either the cell or the surrounding fluid. So when you move the orb, when you try to move the orb, either the body will move or the water will be pushed back. So either the movement of the cell or the surrounding fluid. It will either move the surrounding fluid or the cell itself will move. So can I write or like movement? Can I write ors like movement? Ors like movement. Okay. Flagella is comparatively larger. It is going to be longer. It is much more longer and responsible for the cell movement. It is responsible for the cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different. See, difference again. Are structurally different from that of eukaryotic flagella. So there is a vast difference. There is a vast difference between prokaryotic flagella as well as eukaryotic flagella. Both are very different from each other. They are not same to same at all. Okay. Now, electron microscope study. Students, any doubt till this first paragraph? Any doubt in the first paragraph? Only thing which you need to know here in the first paragraph is the difference between cilia and flagella. Now, the electron microscope study of cilium or flagella shows that they are covered by a plasma membrane. So the outer layer is called as what? The outer layer is called as the plasma membrane. Done. Their core is called as axoneme. Friends, the entire part is called as axoneme. That is the core. This entire thing is called as the core or axoneme. It's called as core or axoneme. Possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules. Point number 1. Can you see here, students? There are in total of 9 peripheral microtubules. Which are doublet here. Okay, which are doublet. A pair of centrally located microtubules. So in the center also there is the pair of microtubules. Centrally located microtubules. Central microtubules. Okay. Now, such an arrangement of axonemal microtubules is referred to as 9+2 arrangement. 9 on the periphery, 2 in the center. Okay. The central microtubules are connected by bridges. Here. Central microtubules are connected by bridges. Okay. Bridges. And is also enclosed by a central sheath. So it is also enclosed. The central microtubules are completely surrounded by a central sheath. You see, this is the central sheath. This is going to be the central sheath. Okay, central sheath. Now friends, which is connected to one of the tubules of the each periphery doublet by a radial spoke. Very important to understand this concept here. See here, each central sheath is connected to the peripheral microtubules. Central sheath is connected to the peripheral microtubules with the help of radial spoke. With the help of radial spoke. So I repeat again, the central sheath is connected. The central sheath is connected to the peripheral microtubules with the help of radial spoke. Radial spokes. So tell me, if there are 9 peripheral protein students, if there are 9 peripheral microtubules, how many radial spokes will be there? Quickly go in the chat. How many radial spokes will be there? Obviously 9. Every single periphery will have one radial spoke. Done. No. Thus there are 9 radial spokes. It's given here. See, there are 9 radial spokes. 9 radial spokes. The peripheral doublets are also interconnected by linker. See, linker, we wrote linker. Peripheral microtubules are connected to each other. The peripheral microtubules are connected to each other with the help of linker. Linker here, linker. Okay, with the help of linker. Both cilium and flagellum emerge from the centriole-like structure. See, they're emerging from the centriole-like structure called as the basal body. So flagella as well as cilia is emerging from the basal body. Emerging from the basal body. See here. This is called as the basal body. They're arising from the basal body. If you look at the structure of your cilia flagella, can you see it is hollow in nature? It is made up of microtubules. I told you, microtubules are hollow in nature. Didn't we learn that? Didn't we learn that microtubules are hollow in nature? They make assembly. Cilia and flagella is made up of microtubules. Tubulin protein, it is made up of tubulin protein. See here, this is called as periphery microtubules. This is radial spoke, central microtubules, central sheet. Done. This entire thing is called as the basal body, through which your entire cilia flagella is emerging. Very easy to understand. If you're not able to understand it the first time, I would advise you to re-watch the session once, then you will have a better clarity. And the reason I'm telling you this is because I want you to draw and learn and not by heart any of this. Very easy to understand. Okay. See here. This is the basal body from which the flagella is arising. These are the microtubules. They are hollow in nature. Covered by the plasma membrane. The inner part is called as axoneme. Okay. Now friends, if there is no doubt, let's go with the next concept now. Can we start? Students, quickly smash the like button, all of you. Quickly mark your attendance and some green hearts in the chat for me to understand that yes, you're understanding the concept. We covered every line of NCERT, students. Every line of NCERT is covered. Now, again, your entire centrosome and centriole is also very easy to understand. Just follow my words. Follow my diagram, and you're good to go. Okay, understand with me. Now, centrosome. Can we draw the structure, students? Can we draw the structure only directly? Let's draw the structure of centrosome. Okay. Now here, in the case of centrosome, students, in the middle, there is no protein actually. In the case of centrosome, in the middle, there is actually no protein. There is no protein at all. In the case of centriole, in the middle, there is no protein. There is no protein at all. Instead, there is no tubulin protein. One, two, three, 4, 5, 6, 7, okay, 8 and 9. Okay, we have a structure here. Now students, in the case of your centrioles, this is a structure of a typical structure of, okay, why does it look so weird? Okay, what, I'll draw it myself. Okay. 3, 4, 5, 6, 7, 8, 9. This looks much more better, no? Yes, this looks much more better. Students, here, in the case of your centriole, in the case of centriole, in the middle, there is something called as hub. In the middle, there is something called as hub. This is the hub. This in the middle, this is the hub. Proteinaceous hub. Proteinaceous hub. Okay. Now here, in the periphery we have myotubules. In the periphery we have myotubules. But here, in the case of centriole, there is no doublet. There is triplet. There is triplet here. There's triplet here. Can you see? 1, 2, 3. 1, 2, 3. Also draw with me. 1, 2, 3. Okay, 1, 2, 3 done. C B A, C B A, C B A, C B A, C B A. Okay, CBA is there. Okay, we have to draw the linkers now. We'll draw the linkers also. There is C2A linker is there. Okay. Now students, this outer part here, this 1, 2, 3, they are called as peripheral microtubules. Peripheral microtubules. Now tell me, are they in doublet or triplet now? They're in the form of triplets. Each different peripheral microtubules are connected to each other. They're connected to each other in the form of C2A linker. C2A linker here. Now, this peripheral microtubules is connected to the central hub. Peripheral microtubule is connected to the central hub with the help of radial spoke. The entire structure is covered by an amorphous structure. From this amorphous structure, we can see spindle fibers. Arising. We can see the spindle fibers arising. Okay. Understand now. Understand the entire structure. When you read, you will understand better. When you read, you will understand much more better. Okay. The centrioles or centrosome is an organelle usually containing two cylindrical structures. First of all, if this is a cylindrical structure, there are two cylindrical structures present at 90 degrees to each other. Okay. This is your entire thing is called as the centrosome. The entire structure, this entire structure is called as centrosome. Individual structure here, which is in a cartwheel structure. Can you see this one? Individual, there are two of them, right? There are two of them. They are called as centrioles. They are called as centrioles. We call them centrioles. The entire structure is called as your centrosome. Entire structure is called as centrosome. Okay, all of you understood. Now students, can you see this amorphous structure around? This amorphous structure. This amorphous structure which is covering the entire centrosome. Entire structurosome is called as your amorphous pericentriolar material. Amorphous pericentriolar. Amorphous pericentriolar material. Amorphous pericentriolar material. Amorphous pericentriolar material. This outer part. This amorphous pericentriolar material is also called as kinoplasm. It is also called as kinoplasm. There are multiple names to it, but just remember what is given in NCERT: amorphous pericentriolar material. Okay. Now understand the lines. Two cylindrical structures called as centrioles. See, we call them two centrioles. They are surrounded by a amorphous pericentriolar material. They are surrounded by amorphous pericentriolar material. Both centrioles in a centrosome are perpendicular to each other. They are perpendicular to each other, in which an organization like a cartwheel. This looks like a cartwheel. They are made up of nine evenly spaced peripheral fibril of tubulin protein. Students, you tell me in the chat now. What is made up of tubulin protein? Is it microfilament? Is it microtubules? Or is it intermediate other one? Yes? Which one is it? Tell me. Obviously, sir, you told us long back. Sir, you told us long back your microtubules are made up of tubulin proteins. Microfilaments, actin and myosin. Intermediate filaments, non-contractile protein. Non-contractile protein. Okay, sir. So technically speaking, they are talking about peripheral microtubules. Peripheral microtubules, which is made up of tubulin protein. It's made up of tubulin protein. Made up of tubulin protein. There, tubulin protein. All of you know now. All of you know. Yes. Now, students, tell me, if there are periphery, there are 9 microtubules here. Periphery, there are 9 microtubules. 9. Plus, in the center, do you see any microtubule? No. I told there is a hub. In the center, there is a hub, but there is no microtubule here. So, can I tell 9 plus 0 arrangement? 9 plus 0 arrangement. In the center, there is a hub. It is not a microtubule. In the center, it is not a microtubule. It is just a hub. Proteinaceous hub. It is proteinaceous hub. So, it is 9 plus 0 arrangement. 9 plus 0 arrangement. Okay. See. Triplets of protein. Each of the peripheral fibril is triplet. The adjacent triplets are also linked. Adjacent triplets are also linked here. They are also linked. Adjacent microtubules are also linked with the help of, not radial spoke, with the help of linker, with the help of Cl linker. With the help of Cl linker. Okay. Now, the central part of the proximal region of centriole is a proteinaceous and called as the hub. Central part is called a hub, which is connected to the tubulins of peripheral. See, tubulins of peripheral triplets by radial spokes. So, the hub is connected to the peripheral microtubules with the help of radial spoke. With the help of radial spoke. Made up of protein. Okay, made up of protein. Right? Made up of protein. The centrioles form the basal body of cilia and flagella. See, centriole forms what? The centriole make up the basal body of cilia and flagella. And spindle fibers that give rise to the spindle apparatus during the cell division in the animal cells. So, the amorphous structure will give rise to spindle fibers during the mitosis and meiosis. Done. That's all. That's all is the difficulty of your microtubules. Very easy it is. Very easy to understand. here. This is like a cartwheel. Can you see? It is a cartwheel-like structure. Perpendicular to each other, it is cartwheel-like structure. It is hollow in the center. Why? Because it is a tubulin protein. It is not tubulin protein. It is a microtubule. Triplets on the outside. The center part is called as the hub. The center part is called as the hub. Microtubules. Triplet. Central part is called the hub. Microtubules are connected to each other with the help of Ca linker. What is difficulty in this? Students, I want you to re-watch this part again. So you can actually understand it if you have any difficulty. Okay? And done. Now students, with that, both difficult parts of cilia and flagella and centrioles is done. Now we'll go towards nucleus. Can we start, students? It's such an easy concept, right? Such an easy chapter it is. Such an easy chapter. Everything is very easy if you understand clearly. Okay? Clear? Students, can we start now? Now students, nucleus, students, you need to know that after this class, right? How many of you in this PYQ session? After today's class, in this video only, in this session only, the entire chapter PYQ will be doing. So you will get a complete revision. So if you're new to the channel, quickly smash the like button, because PYQ part is going to be super exciting. We'll be doing a rapid fire questions. We'll be solving assertion reasons, match the following, fill in the blanks, all types of questions towards the end of today's class. Okay. Now students, can we start with nucleus now? Quickly tell me in the chat, can we start with the nucleus? Very easy again. Very easy to understand nucleus. Now, let's draw it first. Let's try to draw this nucleus. Entire nucleus we can draw. Okay. Let's draw the entire nucleus. Okay. Tell me students, is the nucleus single layer or double layer? Is the nucleus single layer or double layer? Is the nucleus single layer or is it double layer? Okay. Now we have two layers now. Your nucleus, which is a double layer. This is the outer layer. This is the outer layer. This is the inner layer. Outer layer as well as inner layer. Together they make up the nuclear membrane. Together they make up the nuclear membrane. Okay. Outer layer and the inner layer. Now students, between the outer layer and inner layer, there is something here. Can you see? Between the outer layer and the inner layer, there is a structure here. There is some kind of material. Between the outer layer and the inner layer. Between the outer layer and the inner layer, there is something called as perinuclear space. Perinuclear space. Between outer layer and inner layer, there is something called as perinuclear space. Okay. Now, what about the center? In the center, we have some form of matrix. That is called as nucleoplasm. This nucleoplasm is nothing but the matrix. Nice. Now, students, if you remember the structure of, when I was teaching you endoplasmic reticulum, did I not tell you that from the outer layer of nuclear membrane, ER is developing? Yes. Did I mention? Yes, I mentioned. From the outer layer of the nucleus, endoplasmic reticulum is developing. Yes. From the outer layer of ER, endoplasmic reticulum is developing. From the outer layer. Now students, on this endoplasmic reticulum, if I find some ribosomes, Will it be smooth ER or rough ER? Tell me. On this ER, endoplasmic reticulum, if I find ribosomes, can I call this structure RER, rough endoplasmic reticulum? Yes. Can I call this rough endoplasmic reticulum? Yes. Rough endoplasmic reticulum. Nice. Now, students, you need to understand that on this nuclear membrane, the outer membrane, inner membrane, there are small gaps. In this, between the outer membrane and inner membrane, there are small gaps. Between the outer membrane and inner membrane, there are small gaps like this. There are small gaps through which RNA and protein can come out. And these small gaps are called as nuclear pores. They are called as nuclear pores. Through which nuclear pores? Through the nuclear pores, RNA and proteins can come out. RNA and proteins can travel. RNA and proteins can travel through the nuclear pore. Students, always remember, even on the membrane, even on the nuclear envelope, we can find some ribosomes here and there. Even on the nuclear envelope, we can find some ribosomes. Okay. Now, what about inside? What about the genetic material, students? What about the genetic material? The genetic material, will it be in the form of chromosome? If I'm talking about interface, if I'm talking about interface nucleus, if I'm talking about interface nucleus, how do you find the genetic material? Will it be in the form of chromatin, or will it be in the form of chromosomes? Tell me in the chat. It will be in the form of chromatin. Exactly. It will be in the form of chromatin. Exactly. This will be chromatin. All the genetic material will be in the form of chromatin. This is the chromatin. That is going to be a chromatin. Which is basically the DNA here. Now, students, in the nucleoplasm, there are some special structures. In the nucleoplasm, there are this round structures. There are some amorphous structures are there. Amorphous structures are found in the nucleoplasm, the matrix. In the matrix, we can find some round structures which are present in the nucleoplasm. These structures are called as nucleolus. They are called as nucleolus. The main function of nucleolus is to produce rRNA. To produce the rRNA. Done, students. This is the entire structure. This is the entire structure of the nucleus. Let me recap one more time. This is the nucleus. It is a double membrane structure. Outer membrane, inner membrane. Together they make up the nuclear membrane. Inside part is called as the matrix, and the name for the matrix is nucleoplasm is the matrix name, nucleoplasm. In the outer layer, we find the development of rough endoplasmic reticulum if it has ribosomes. Ribosomes can also be found on the nuclear envelope. The space between outer membrane and inner membrane, is called as perinuclear space. It is called as perinuclear space. In a perinuclear space, we can find small gaps or the holes. In the perinuclear space, we can find small gaps or holes. These small gaps or holes are called as nuclear envelope. They are called as nuclear pores. They are called as nuclear pores. Now, in the nucleoplasm, within the nucleoplasm, we can find the genetic material. The genetic material is in the form of chromatin. Okay. Now, inside the cytoplasm, inside the nucleoplasm, we find something called as nucleolus. Nucleolus. The main function of nucleolus is to produce rRNA. To produce the rRNA. Done. Do you realize entire structure of nucleus is covered in one diagram? In one diagram. Let's read what's given here. Nucleus as a cell organelle was first described by Robert Brown. All of you know this. All of you know this. Robert Brown. Now tell me, students, who was the first scientist to observe the living cells? Quickly in the chat. Who was the first scientist to observe the living cells? Quickly go in the chat. Okay. As early as 1831. Later, the material of the nucleus stained by basic dye was given the name chromatin by Fleming. Chromatin was discovered by the Fleming, which I'll talk later on. Euchromatin, heterochromatin is there. The interphase nucleus of when it is not dividing, basically the resting stage. Interphase, the resting stage one. Okay, has highly extended and elaborate nucleoprotein fibers called as chromatin. We can find the chromatin inside. Nuclear matrix and one or more special bodies called as nucleoli or nucleolus, which we showed. Okay. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membrane, so double membrane, with a space between, the between space is called as perinuclear space. Perinuclear space. Okay, perinuclear space forms a barrier between the materials present on the inside of the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum. See, outer membrane, inner membrane. Middle layer is part called as perinuclear space. Outer membrane has ER continuous, ER. Inside part is called as chromatin, which has the genetic material with the endoplasmic reticulum, and also bears ribosomes on it. See, I told you, even on the ribosomes are present on the ER. At a number of places, nuclear envelope is interrupted by minute pores. The minute pores are called as nuclear pore, which are formed by the fusion of two membranes. So very important line. So how do you get nuclear pore? In the nuclear pore is formed by the fusion of two membranes. Outer membrane, inner membrane, they fuse together and they make the nuclear pore. These nuclear pore are the passage through which the movement of RNA and protein molecules takes place. So movement of RNA and movement of protein with the help of the nuclear pore. Nice. Normally, there is only one nucleus per cell. Usually, there is only one nucleus per cell, but it can vary. Some of the cells can have multiple nucleus. The syncytium condition, the free nuclear endosperm. Yes. Variation number of nuclei are frequently observed. Okay. Some mature cells even lack nucleus. For example, RBC cell, red blood cells, erythrocytes, there is no nucleus. It is just the cytoplasm and the outer layer. That's it. RBC. Of many mammals and sieve tube cells, which are completely hollow, they do not have any nucleus. The main function is conduction. Okay. Now tell me, the sieve tube, do you find in xylem of phloem? Quickly go in the chat. Xylem of phloem. Okay. Now, the nuclear matrix of the nucleoplasm contains nucleolus and chromatin. Nucleoli are spherical structures present in the nucleoplasm. The contents of nucleolus is continuous with the rest of the nucleoplasm. That's why it is very amorphous in nature, as it does not have any membrane bound structure. Okay, it does not have any membrane bound structure. So nucleolus does not have any membrane. It is a site for active ribosomal rRNA synthesis. Ribosomal RNA synthesis. That is nucleolus or nucleoli. Okay. Large and more numerous nuclei are present in actively carrying out present in the cells actively carrying out protein synthesis. That is based on the requirement. For example, a cell, if the cell requires more protein, there will be more nucleoli. More protein synthesis, more nucleoli in that cell. Simple it is. More protein requirement, more nucleoli. Done. With that, this part is also done. Now students, now can we learn about chromosome now? Can we shift to chromosome? Very smooth class is going. The class is going very smooth. Every single concept which is said to be difficult is been made easy very easily. Right? Every single concept. Now we'll go to the next concept, that is going to be the structure of chromosome. Structure of chromosome. Ready? Ready students? Before we go to structure of chromosome, I want you to understand the difference between chromatin. Understand difference in chromatin. Okay, understand. There's something called as euchromatin. Euchromatin and heterochromatin. Euchromatin as well as heterochromatin. Very simple difference, understand very easily. Understand very easily. Okay. Now let me show you a demonstration. Students, if this is your entire chromatin, this is your chromatin, students. This is your chromatin. This is your chromatin. Sometimes chromatin is very tightly bound. You see, sometimes the chromatin can be very tightly bound. Sometimes the chromatin can be very loosely bound. There is no loose, very loose. When the chromatin is loosely bound, can you see? When the chromatin is very loosely bound, it is called as euchromatin. When it is loosely bound, it is called as euchromatin. When it is loosely bound, it is called as euchromatin. When it is tightly bound, it is called as heterochromatin. It is called as heterochromatin. Now tell me, on this chromatin, students, on this chromatin, if I take this chromatin and if I add stain on top of it, if I take this chromatin and I add stain, tell me which one will be darkly stained? Will this knot will be darkly stained, or this small loose chromatin will be darkly stained? Tell me, which will be darkly stained? Obviously, students, this entire structure, which is the tightly bound structure, darkly stained. Yes, it will be densely stained. Densely stained. This is going to be lightly stained. Lightly stained. Yes, lightly stained. Now tell me, if I take this entire chromatin and if I want to make RNA, out of it, which will be easy? If I have to take this entire thing, I have to unwind it and then make RNA. If I have to take this simple loosely bound chromatin and if I have to make the RNA, which will be more easier? Obviously, this one will be more easier, right? Compared to this knot, this will be much more easier. Yes. So can I tell students, your euchromatin is going to be transcriptionally, it is going to be transcriptionally active. It is going to be transcriptionally active, while your heterochromatin is going to be inactive in nature. It is going to be inactive in nature. Done. That's all. That's all the difference between euchromatin and heterochromatin. Euchromatin as well as heterochromatin. Okay. Euchromatin as well as heterochromatin. Once again, students. Okay. Euchromatin as well as heterochromatin. Now, students, I want you to understand the structure of the entire chromosome now. Entire structure of chromosome. Okay. If you look at the structure of chromosome, if you look at the structure of a typical structure of a chromosome, in the middle we have a primary constriction. In the middle we have a primary constriction. Primary constriction. This primary constriction in the middle is also called as centromere. It is also called as centromere. It's called as centromere. Okay. Primary constriction is called as centromere. Now, students, remember this primary constriction, which is centromere, is said to be inactive in nature. It is said to be inactive in nature. Inactive. Now, if it is inactive in nature, which type of chromatin do you think it is? It is made up of which type of chromatin? Tell me. Then heterochromatin. It is made up of heterochromatin. Heterochromatin, because it is transcriptionally inactive in nature. Transcriptionally inactive in nature. Okay, inactive in nature. Okay, primary constriction done. Now, students, this entire structure of your chromosome has two chromatids like this. It has chromatids. It has chromatids. They are called as sister chromatids. This one and this one. This one and this one, they are called as sister chromatids. They are called as sister chromatids. They are called as sister chromatids. Sister chromatids. Okay, they are called as sister chromatids. Done. Done. They are called as sister chromatids. Here, extra information. This is called as the P arm. This is called as Q arm. Just remember that. P arm as well as the Q arm. Remember that. Okay. The end points now. End points. The end points of the chromosomes or the chromatids. Can you see? Chromosomes or chromatids are called as telomeres. They are called as telomere. They are very important. They protect they completely protect the chromosomes. Protect the chromosomes. They are called as telomeres. Now, students, on the top here, on the top, we find the secondary constriction. Can you see? On the top here, we find the secondary constriction. We find the secondary constriction. This secondary constriction is also called as satellite. It is also called as satellite. It is also called as satellite. But unlike your primary constriction, the secondary constriction is going to be active in nature. It is going to be active in nature. And also remember the out of 23 pair of chromosomes, 23 pair of chromosomes, 5 of them will have the constriction, will have the secondary constriction, Remember that point. Now, students, last point. Around the primary constriction, there is a protein layer. Around the cup-shaped protein layer around the primary constriction. The cup-shaped protein layer around the primary constriction is called as kinetochore. This is called as kinetochore. It's called as kinetochore. It's called as kinetochore. And always remember, students, during the cell cycle, during the cell division, okay, cell division, chromosome splitting, centromere splitting, or the homologous chromosome splitting, the point of attachment of spindle fiber, to the chromosome is always at the kinetochore. All the spindle fibers will always attach. The spindle fibers will always attach at the kinetochore. They are the spindle fibers. Spindle fibers will always attach at the kinetochore. That's how it works. Understood? Any doubt you quickly ask me. Any doubt here, quickly ask me, students. This is the structure of your chromosome. This is the entire structure of chromosomes. Entire structure of chromosomes. Done. Structure of chromosome done. Let's read what's given here. Okay. Let's read what's given here. You may recall that interphase nucleus has a loose and distinct network of nucleoprotein fibers called as chromatin. Yes, we know chromatin. Okay. But during the different stage of cell cycle, the cells show chromosomes. Obviously, during the after prophase, metaphase, anaphase, the chromosome will show a proper shape. The chromosome will show a proper shape. In the place of nucleus, chromatin contains DNA and some basic proteins called as histone proteins. Very important. So your chromatin will have the chromosomes will have the chromatin contains DNA and some basic proteins will be there, that is called as histone protein. This histone proteins which you learn in molecular basis, the octamer protein, H2A, H3, H4, H2A, H2B, all that you learn in molecular basis of inheritance. Remember right now, chromatin has what? Chromatin contains DNA and histone and some non-histone proteins and also RNA. So if they ask you what does chromatin contain, chromatin contains DNA, histone, non-histone as well as RNA. 4 points, all of you. A single human cell has approximately 2-meter long thread of DNA distributed among the 46 chromosomes. Okay. Every chromosome visible only when the dividing cells essentially has a primary constitution called as centromere on each side of the disc-shaped structure called as kinetochore, where the spindle fibers attach. Centromere holds the two chromatids on the chromosomes. Based on the position of centromere, students, very important line. Based on the position of the centromere, where the centromere is at, there's a primary constriction. We have different types of shapes of chromosomes. Okay, chromosomes can be classified into 4 types: metacentric chromosome, submetacentric, acrocentric, as well as telocentric. Can you see here? You can see here. Metacentric, submetacentric, acrocentric, and telocentric. Based on the position of centromere, it can be divided into metacentric, submetacentric, acrocentric, and telocentric. Here, almost. Metacentric is V shape, metacentric is V shape, your submetacentric is going to be L shape, acrocentric is going to be J shape, and telocentric is going to be I shape. That is VLJI. That is VLJI. Can you see? VLJI. VLJI shape. VLJI shape. Okay. VLJI shape. Clear, students? Your metacentric is going to be always going to be V shape, submetacentric L shape, acrocentric J shape, and telocentric is going to be I shape. Based on the position of the centromere, we can see the different shapes of chromosomes. Different shapes of chromosomes. Very easy. Very easy. VLJI. 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 Repeat after me, all of you. VLJI. That's how you can remember. That's how you can remember. VLJI shape. Okay. VLJI shape. Some, we didn't read here. Huh? See here. Metacentric chromosomes has middle centromere forming equal arms. P and Q arm are equal in metacentric. Submetacentric chromosome has centromere slightly away from the mid of the chromosome, resulting in one short arm and other longer arm. Can you see here? In the case of submetacentric, what is happening? Your Q arm is going to same. POR is going to remain the same, but the P arm is going to be short. The P arm is going to be short. Now, in the case of your acrocentric, it is almost going towards the terminal. In telocentric, full terminal. Telocentric, full terminal. Very easy. Okay. See here. Submetacentric chromosome has, okay, acrocentric chromosomes, the centromere is situated very close to the end, forming the extremely short and one very long arm. Whereas the telocentric chromosome has a terminal centromere. Telocentric, terminal centromere. Telocentric. Telocentric is going to be terminal centromere. Telomere, VLJI. Sometimes a few chromosomes have non-staining. Okay. If it is non-staining, can I also call it light staining? Can I tell as it as light staining? Light staining. If it is light staining, students, if it is light staining and it is going to be transcriptionally active, can I tell this is a type of euchromatin? Type of euchromatin, yes. Non-staining. Secondary constriction at a constant location give appearance of a small fragment called as satellite. That is going to be your satellite. Light staining. Microbodies, small concept. Many membrane-bound minute vesicles called as microbodies. Microbodies are present inside the eukaryotes. Microbodies that contain various enzymes are present in both animals. Microbodies. That's all. And with that, the chapter is over. With that, the chapter is over. Easy, right? Such an easy chapter, students. Such an easy chapter. Now, students, I'll give you a small glimpse. On the right side, can you see? On the right side, can you see number of questions? On the right side, can you see number of questions? Questions are never ending. Questions are going to be never ending. Can you see? So many PYQs for today's class. So many PYQs for today's class. You see? Never ending questions are there. Never ending questions are there for today's class. Never ending questions there for today's class. Now, do you want to solve the PYQs today? Yes or no in the chat, first of all. Do you want to solve those PYQs today? Because once you do a chapter, if you solve the PYQs, then only there is a meaning for the chapter. Because when you learn something, if you want to know that, yes, I have learned something and applied, you solve questions. Right? You solve the questions. Okay? Now, students, we will be solving this question because I told you other day in Telegram I will add the questions, but I could not. So that is why I am doing this. That is why I bought the PYQs. Because that day I told you in my Telegram I will add questions, but I could not add. Right? That's why in today's class I bought so many numericals, so many questions. Numerical session this week, this Saturday or Friday I'm doing. This Friday or Saturday we are doing numericals. Okay? This Friday or Saturday we'll be doing numericals. Because that is RNA series, right? This last week I was not here, right, in Bangalore. So that we'll be doing. Okay. So students, can we take a 20 minutes break because I want to relax my throat because I've been teaching from morning. This is my third class for the day. Paid batches and YouTube, both way I take. So can I rest my throat for some time and for 20 minutes and then can we start? Sony, Mona, don't worry. This week we are doing numerical session. Okay. Okay. Now time is exactly 6:22. 6:42 will start. 20 minutes break. We'll take a small 20 minutes break. I want everyone to relax also for some time and then we will start with the questions. 6:42. Okay. 6:42 will start. Okay. 6:42 will start. 20 minutes break will take. Aramsi will take. We'll come back after 20 minutes and then we'll start. Okay. I want you to relax yourself, take rest and then we'll start. 6:22 class will resume again. Okay. And we'll be solving a lot of questions. Question time. Until then, bye-bye. Okay, I'll come back. Okay. So hello students, welcome back. Hello. Yes, welcome back to channel now. Students, tell me in the chat if you're ready for solving questions now. Yes, are you ready to solve the questions now? Because questions are going to pop up. I'll give you a solid 30 seconds or 20 seconds for every question. Then we'll start solving them one by one. What's it? Okay? Can we start, students? Can we start? Quickly, let me know in the chat if you're ready for the PYQ time now. PYQ time. Okay? Ready? Yes. Students, quickly smash that like button and share the video to everyone. Quickly smash that like button and share this video to every single one of the students, because it is very important for you to spread the word, right? Very important for you to spread the word, because no one is doing at this point. We are covering every single line of NCERT. We're doing PYQs, every single thing at one point. Okay, so let's start with the PYQs. The first one is here. NEET 2024 question. Go. Match the following: nucleolus, centriole, leucoplast, Golgi apparatus. So we just learned that nucleolus is the site for ribosomal. So A is going to be 3. If A is going to be 3, can I tell option number 1 and 3 is gone? 1 and 3 is gone. Okay. Now centrioles. Centrioles is the cartwheel-like structure. Today only we learned. So B is going to be 2. A is going to be 3 and B is going to be 2. Only one option is there. Easy now. Time starts now. Go quickly. If A is going to be 3 and B is going to be 2. Go. What is the answer? What is the answer? Quickly, let me know in the chat. I'm going to move aside so all of you can read the question very well. Okay. Obviously, it's going to be option number D. So, nucleus, we just now learned today's class. Nucleus is the site for your ribosomal synthesis. Centriole is going to be the cart-like structure. Leucoplast is for storing the proteins, and Golgi is for glycolipids. Now, Golgi apparatus, does it produce only glycoproteins or also glycolipids? Both. Glycoproteins and glycolipids, both are produced. Okay. Next question. The DNA present in chloroplast is? Is it going to be single, double stranded, linear, single stranded, circular, single stranded, linear, double stranded? Students, remember, the DNA which is present in the case of chloroplast is similar to the DNA which is present in the case of bacteria. Tell me. Absolutely correct. It is going to be circular and double stranded. It is going to be circular as well as double stranded. Nice. Very easy question. Next question. Very simple again. Understand. Axoneme. First of all, tell me, where did we study axoneme? We studied axoneme in cilia and flagella. The entire core in the 9+2 arrangement of cilia, we find axoneme. So A is going to be 2. What about B? Cartwheel pattern, that is centriole. So A is going to be 2, B is going to be 1. Okay. Cristae, we find cristae in the case of mitochondria, and satellite, which is also called as the secondary constriction, is found in the case of chromosome. So what is the answer now? Easy. Option number 3. That is axoneme is part of your cilia and flagella, cartwheel, centriole, cristae, mitochondria, satellite is the chromosome. Easy. Yes, very easy. Next question. The cell wall formation in bacteria is facilitated by cell wall formation. It is not just cell wall formation, it is also DNA replication, distribution of daughter cells, increasing the surface area, enzymatic activities. All of these functions are a part of one structure. That is, if this is the layer, this is going to be the structure, outer structure, then we have middle structure, then we have inner structure. From the plasma membrane, there is some constrictions, there are some extension of plasma membrane. The extension of plasma membrane are called as mesosomes. They are called as mesosomes is the answer here. Mesosomes. Easy? Very easy. Next question. Which of the following represents sister chromatids in the given figure? Sister chromatids. Students, both this center part A is centromere. What about the C? C is one chromatid. C is going to be one chromatid. Okay. What about D? D are non-sister. So what about B? B is going to be sister. B is going to be sister chromatids. So what is the answer? Harshini, Harshita, Meghana, Madhumita, Paul, Ravindran. Yes. Answer number B. Answer number B. And students, do you realize this is a NEET 2024 questions? Don't you think so? The questions were very easy. Don't you think so? After reading the NCERT line by line, you are able to solve every single question very easily. This is the power of the NCERT line by line. NCERT line by line. Next question again. Next question. Cilia. Tell me. Cilia and flagella, I told you, axonym. Right. Endoplasmic reticulum as ribosomes. Mitochondria as cristae. Yes. Kinetochore where the spindle fibers attach. Tell me the answer now. Tell me the answer quickly. Go. Cilia is going to be axonym. Yes. A is 3. A is going to be 3. B is going to be 4. B is going to be 4. C is going to be 2. And D is going to be 1. Option number 2. Option number 2. Quickly. What is the answer? Cilia is going to be axonym. Endoplasmic reticulum is going to be ribosomes. Mitochondria is going to be cristae. Yes. Option number 2. Very easy. Very easy again. Next question. Next question. Again from 2024. Okay. The Golgi cristae are concentrically arranged near the nucleus. Okay. With distinct convex cis or the forming phase and the concave trans or maturing phase. Okay. The cis and the trans phase of the organelles are identical and interconnected. Students, the cis and the trans phase, are they interconnected? No. Cis phase this side, the forming phase. Trans phase is the other side, called as the maturing phase. Cis phase, it is entering. Trans phase, the vesicles are coming out. They are not connected to each other. They are not connected to each other. So option number 3. Option number 3. Okay. Option number 3. A is true. B is going to be false. Okay. Clear? Next question. Which of the following functions is called out by cytoskeleton in the cell? In today's class we did this. In today's class we did this. Today's class we did it. Today's class we did it. Function of cytoskeleton. Transport, protein, nuclear division, motility. See, cytoskeleton, the main function was support. First one. Second one was something which is given here. The third one was it provides the motility, and we have the structural support. The shape, support, and motility. Okay. Next question. Which of the following statements concerning the endoplasmic reticulum is going to be incorrect? It is going to be incorrect. Time starts now. Incorrect statement. Okay. SCAR are a site for lipid synthesis. Yes. Smooth endoplasmic reticulum, lipid synthesis. Rough endoplasmic reticulum, ribosomes are attached. Yes. SCR is devoid of ribosomes. Yes. In smooth endoplasmic reticulum, there are no ribosomes. Only in rough endoplasmic reticulum, there are ribosomes. Okay. In prokaryotes, only RAR is present. Absolutely not. In the case of prokaryotes, we do not find endoplasmic reticulum. There is no endoplasmic reticulum at all. Right? So option number 4 is incorrect. Now, next question. The membrane-bound organelles of endomembrane system coordinate cellular function. Yes, they coordinate cellular function. That's why they are called as endomembrane. Mitochondria and chloroplasts are not considered a part of endomembrane system. Obviously, they are not a part of endomembrane system. Quickly, what is the answer? Hello, Shreya. What is going to be the answer? Quickly, let me in the chat. What is going to be the answer? Okay. First one is correct, second one is correct. So obviously, both statement 1 and 2 are correct here. Both 1 and 2 are correct here. Okay. Both 1 and 2 are correct. Okay. 1 second, students. 1 and 2 are correct. Okay. Next question. Okay. Very simple, match the following. Very simple, match the following again. Very simple, match the following. Chromoplast, amyloplast, ileoplast, alveoloplast. Chromo provides color, carotenes. Amylopectin is a type of starch. Ileoplast, oil. Aileron layer, protein. So what is the answer? Tell me in the chat now. What is the answer? Quickly, go. So A is going to be 4. B is going to be 3. C is going to be 2. D is going to be 1. So what is the answer? I moved. What is going to be the answer? Think, Harshita, think. Chromo is color. I'll erase everything. See. Chromo is color. Okay. Amylopectin is a type of starch. Ileoplast, oil. Aileroplast is aileron layer, proteinaceous layer. So 2 is the answer. Easy. Okay. Done. Next question. Which type of substances would face difficulty in passing through the cell membrane? Some of the molecules cannot enter. So remember, students, this is a small trick. Not trick, you should know this, and we should know this. Through a cell membrane, if you look at here, if you look at the structure, if you look at this definition here, students, here it says that neutral solutes may move across the membrane by process of simple diffusion. Okay. Polar molecules cannot pass through the non-polar bilayer. They cannot pass through. Polar molecules are also hydrophilic in nature. They are going to be hydrophilic in nature. Yes, they are going to be hydrophilic in nature. Polar molecules are hydrophilic in nature. So what is the answer now? Tell me. Substances which are hydrophilic moiety are going to be hydrophilic moiety. 2 is the answer. Easy question. Okay. Next question. If the pH of lysosomes is increased to alkaline, that is, if the pH is going to be increasing, what will be the outcome? Hydrolytic enzymes will function more efficiently. Hydrolytic enzymes will become inactive. Lysosomal enzymes will be released into the cytoplasm. Lysosome enzymes will become more active. Go. Tell me quickly. Tell me. Students, remember, lysozymes, all the enzymes are able to work very well because in acidic condition. Now you take out the acidic condition, what will happen? Tell me. Your hydrolytic enzymes work best at the acidic pH. They work best at acidic pH. Now if you remove the acidic pH only, you make it alkaline, will they work properly? Yes or no? Tell me in the chat. Hydrolytic enzymes work best in acidic pH. They work best in acidic pH. If you remove the acid only, will they become active or inactive? They'll become inactive. Yes, they'll become inactive in nature. Did all of you understand this question now? Understand? Yes, Meghna? They'll become inactive. The enzymes will work better at acidic pH. If you remove the acidic pH only, will they function? Though they'll not function. That's all. That's all is the question. Understood? Yes or no? Quickly. Understood? Nice. Friends, quickly smash that like button if you're new to the channel, because making all these questions take a lot of time. And it is very important for you to know that yes, We are here working very hard from morning till night, and it is fruitful for everyone. Okay. Now, friends, again, simple question. Cristae, thylakoid, centromere, and cristerne. Time starts now. Go. Match the following. You need to understand. You will get a lot of questions. Match the following from here. Meghna, understand. Hydrolytic enzymes in the lysosomes. Hydrolytic enzymes in the lysosomes will work better in acidic pH. They will work better in acidic pH. If you make it alkaline, will the enzymes work? No, enzymes will not work. That's what is the question. Enzymes will become inactive in alkaline pH. Understood? Enzymes will become inactive in alkaline pH. No. Quickly. Done. Cristae, foldings of mitochondria. Thylakoids. Thylakoids is going to be your part of your chloroplast. Centromere, primary constriction. Cristerne, in part of your Golgi complex. So A is going to be 3. So first only 1 is given. Lol. Gone. First is the answer. 1 is the answer. Next question. Which of the following is an incorrect statement? It is an incorrect statement. Time starts now. Go. The perinuclear space forms the barrier between material present on the inside of the nucleus and the cytoplasm. Okay. Nuclear pore act as a passage for protein and RNA molecules in both directions between the nucleus and cytoplasm. Okay. Mature sieve tubes, elements possess conspicuous nucleus and usual cytoplasm organelles. Microbodies are present in both plant and animal cells. Quickly. Which one is incorrect? Quickly. Incorrect statement. Students, do you remember one line of the NCERT? When we were doing NCERT line here, when we are doing the line here of nucleus, when we are doing this one question of nucleus right here, did I tell you this line here? Can you see this line here? Can you see here? It said that erythrocytes of many mammals and sieve tubes of cells of vascular plants, they do not have nucleus. Nucleus is absent. If nucleus is absent here, can you see phloem that it is completely empty? In the case of nucleus is absent in the case of RBC also. Yes. And the statement here says, students, everything is from NCERT if you let it. Okay. The statement here says that mature sieve tube elements possess a nucleus. Do they have nucleus? They do not have nucleus. Simple answer. Incorrect statement. Done. Easy, right? Easy. Next question. Organelle that are included in the endomembrane system. Go. Easy question again. Organelles are part of endomembrane system. Which are a part of endomembrane system. Quickly. Organelles which are part of endomembrane system. Yes. Students, remember the easy trick. What is the trick? The trick is going to be every gamer loves victory. Endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Option number 4. Next question. Which is the important site for formation of glycoproteins and glycolipids in eukaryotic cell? Bacha question, laddu question. Easy answer. That is going to be your Golgi complex. That is going to be your Golgi bodies or Golgi complex, which make up the glycoproteins and glycolipids. This glyco is basically adding the sugar. This protein right here, protein and lipids are synthesized by the endoplasmic reticulum. Endoplasmic reticulum are producing the lipids and proteins. That is, packaging is done by Golgi body. Okay. Next question. Inclusion bodies of green, blue, purple, and green photosynthetic bacteria are. Inclusion bodies in blue, green, and purple sulfur bacteria is going to be your gas vacuoles. Contractile vacuole in the case of your amoeba. Okay, gas vacuoles. Because of the gas vacuoles, the bacteria can show buoyancy. Next question. The biosynthesis of ribosomal rRNA occurs. Very, This is the most repeated question and the easy question which we have been doing till now. That is going to be your nucleolus. Nucleolus is the site for rRNA. Site for RNA synthesis. Next question. The concept of omnis cellula e cellula regarding cell division was first proposed by omnis cellula e cellula. The new cell is arising from the pre-existing cell. The new cell is arising from pre-existing cell was given by Rudolf Virchow. The other two scientists are Scheiden and Swan. Scheiden and Swan along with Rudolf Virchow, together they gave the cell theory. Now, the short arm and the long arm of submetacentric chromosomes are, I told you already, in the submetacentric, submitting from that, this is the P arm, this is the Q arm. P arm and Q arm. Okay, P arm and Q arm. Next question. Which of the following pair of organelles do not contain DNA? That is, DNA is absent. Nuclear envelope and mitochondria. Mitochondria has DNA. Mitochondria and lysosome. Mitochondria has DNA. Chloroplast and vacuoles. Chloroplast has DNA. Lysosomes and vacuoles, this is the right answer. Both of them do not have DNA. Easy answer. Students, do you realize every single line of NCERT they have given you a question. Next question. Which of the following statement is not correct again? Is not correct. Lysosomes are formed by the process of packaging of endoplasmic reticulum. Lysosomes have numerous hydrolytic enzymes. Hydrolytic enzymes of lysosomes are active under acidic pH. See, I told you, they're active under acidic pH. If you decrease the pH, if you increase the pH, they become inactive. Lysosomes are membrane-bound structures. Which is the incorrect statement? Obviously, the first one, because packaging is done by Golgi body, not by endoplasmic reticulum. Packaging is always done by the Golgi complex. Right? A is the answer. Okay, done. That's all, students. That's all. We solved around more than 30 PYQs in the span of 15 minutes. 30 PYQs in 15 minutes. Not 30, I think about 25 PYQs in 15 minutes. It shows you that questions are easy actually here. Questions are easy. It is only a matter of practice. Questions are always going to be easy here. And if you think they are going to be easy, it is because you know the answers with the help of practice which you have done. And students, if you want to continue the line by line series, see, I gave you this promise, so I gave you these questions. Okay. So students, if you're not a part of DNA batch, still, again the price is back to 399, test series 499, Tatva books test series and DNA 7999. Okay. So with that, the chapter is over. With that, the entire chapter is over. Do you want to solve more questions? I can ask you orally also. It's a very easy thing. It's a very easy thing to ask the questions orally. Okay, let me ask you a question. In submetacentric, what is the shape of the chromosome? Simple. In submetacentric, what is the shape of the chromosome? Tell me. Submetacentric, shape of the chromosome. Submetacentric, quickly. Submetacentric is going to be L shape. Done. Submetacentric is L shape. Metacentric, V shape. Okay. Submetacentric, V shape. Now I'll ask you one more question. What is the layer called as? Between the outer layer and inner layer of the nuclear membrane, what is the middle layer called as? Quickly, go. What is the middle layer called as? J shape is going to be in acrocentric. Telocentric is I. VLJI. It is always VLJI. Okay. What do you call the space between the two outer membrane and inner membrane of your nuclear envelope? That is called as perinuclear space. Okay. Perinuclear space. In centriole, what is the arrangement of microtubules? Tell me. What is the arrangement of microtubules in the case of centrioles? Is it 9 plus 1, 9 plus 2, 9 plus 3 or 9 plus 0 in centrioles? Quickly, go. In centrioles. It's going to be 9 plus 0 arrangement in 9 plus 0 arrangement in the case of centrioles. 9 plus 0 arrangement. Now what about the arrangement in the case of your cilia and flagella? In the case of cilia and flagella, what is the arrangement? Tell me. It is 9 plus 0. 9 plus 0, not 9 plus 1. 9 plus 0. 9 plus 0 in the case of centrioles. 9 plus 2 in the case of cilia and flagella. 9 plus 2 in the case of cilia and flagella. Cilia and flagella, 9 plus 2. Okay, 9 plus 2 in cilia and flagella, 9 plus 0 in the case of centrioles. Okay. Now, what is the shape of centriole arrangement? They are perpendicular to each other, 90 degree. What is the arrangement? How do they look like? They look like a cartwheel. They look like a cartwheel. Okay. In the case of eukaryotic ribosomes, which type of subunit do you find? Which type of subunits do you find in the case of eukaryotic ribosomes? 60S and 40S. Together 60S and 40S, that is ATS ribosome. ATS ribosome. Okay, ATS ribosome. Beautiful. Easy question again. Now students, what does the S stands for? What is the S stands for in the ATS or 70S? ATS or 70S, what is the meaning of S? S is going to be sedimentation coefficient or Sedberg unit, also called as Sedberg unit. Nice. Easy question. Now, next question. In the case of what is the core of cilia or flagella called as? What is the core of cilia or flagella called as? The core of cilia or flagella, that is called as axoneme. It is called as axoneme. Okay, it's called as axoneme. Okay. Now, how many types of cytoskeletons do we have in a cell? There are three types. Right? One is called as microfilaments, other one is microtubules, and a third one is called as intermediate ones. Okay, three types. Okay, next question. Let me think. Next question. In the case of chromosomes, we have two arms. What are the two arms called as? What are two arms of a chromosome called as? P arm and Q arm. P arm as well as Q arm. P arm and Q arm. Nice, easy question. What is the secondary construction called as? What is the name of secondary construction? And is it transcriptionally active or inactive? Is it going to be transcriptionally active or inactive? The secondary construction is called as satellite, and it is going to be transcriptionally active. Transcriptionally active in nature. Easy question. Now, spindle fibers are attached to chromosomes. Spindle fibers are attached into the chromosome, but which part of the chromosome? Do they attach to centromere? No. Do they attach to the kinetochore? Yes. They attach to the kinetochore. Nice. Clear? They attach to the kinetochore. Done. Students, if I stand here, I can ask you questions, like around 100 questions will keep on coming. I don't require, because the reason is because I know NCERT line by line and you know NCERT line by line. I can keep on asking questions, you can keep on answering the questions. That is the beauty of NCERT line by line. Do you realize? And with that, I think we should we'll stop the class now. Yes. And let me know in the comment section which is the next chapter you want line by line. Let me know in the comment section which you want line by line next chapter. I was thinking cell cycle and cell division. We will do cell cycle and cell division because then we can finish one entire unit of your cell. Right? Biomolecules I'll do later on. Okay. Clear students? So with that, thank you so much students for today's class, and I enjoyed it. I really enjoyed today's class. It was very fun because many students who do not understand the concept of cilia and flagella, you know the structure now. You know the structure of centriole now. You know the structure of nucleus. You are able to solve so many questions also. Okay. So let me know in the comment section. How was today's class, because I enjoyed it. It was really fun. It was a very chill session. I enjoyed the class, every single bit of it. And similarly we will do every single chapter of your entire 11th and 12th line by line NCERT. Only if you show the Josh. If the Josh is not there, we will stop it. If there is no Josh, we will stop it, because if you do not show the interest, if you do not show the dedication from your side, our side is meaningless then. Our side is meaningless. Okay. So with that being said, thank you so much, students, and have an amazing night, and good night to all of you. Bye, all of you. Bye, bye. Take care. Have a great day.